Okay, welcome everyone to the Air-Sea Fluxus from Space webinar series. Today we have Francesco Davidio, who's going to talk about Lagrangian biogeochemistry in the SWOT era. So I'll let you take it away, Francesco.
Okay, let me share the screen. Okay, can you see my screen?
Perfect. Yes.
Okay, good. So thank you a lot for this invitation to Christine and Sarah. So I saw that this is a meeting especially for people interested in Air-Sea interaction. In my talk, there will be just as a bit of air, but much more sea, in fact. And the focus will be on the fine scales, or maybe I reduce this thing. Okay, so this should be good to see the entire screen. So I divided the presentation in three parts. There will be a first one in which I will speak about the fine scales that I think you all more or less know, but I will especially talk in terms of Lagrangian dynamics. I will make some biogeochemical examples of how Lagrangian methods can be used to explore this beautiful regime, and then I will speak about the limitations of current observational systems in terms of fine scale and great perspectives that the new SWOT satellite mission can bring to this field. So first of all, the approach that I during the talk is a kind of a landscape or a seascape view of the ocean. I like to think to the fine scale studies in a way that can be borrowed from the way in which we look at a land. To terrestrial landscape. We are in fact animals that have evolved on land, and so we have our senses that are that can easily capture some feature of a terrestrial landscape and identify some coherent spatial domains. Then, when we look at the sea, we are charmed by all this moving environment, but for us it's very difficult to detect regions that can be different from the others. The first impression, that is something that it has remained in oceanography for until about one century ago, was to see an environment that was well mixed because of the movement of water. Well, we know now that this actually is due to our biological limitations, and if we look at the ocean with then we see a landscape that is as contrasted as a terrestrial one that is emerging. So this is an image from the Sentinel-3 satellite of ocean color. So the color code is not very important. Red is means strong levels of surface chlorophyll, about 2 milligram per meter cube. Blue values are to zero. What's interesting here is the presence of these water patches of the size of 100 kilometers for the case of mesoscale eddies, and then to few kilometers of thickness for the case of filament that actually are coherent in time with lifetime of days to weeks. So these features are very energetic, they are aware of kinetic energy, and so are characterized by strong frontogenesis. This means that they are associated to strong contrast in water properties. So much like the landscape that the difference, the environmental differences of with respect to a valley. But then their specificity is that they are very dynamical. So this lifetime that goes from days to weeks are much shorter than the lifetime of geological features. And in the ocean, the variability of the fine scale landscape is actually in temporal resonance with biological processes. If we think, for instance, to demographic time scales of phytoplankton, or the temporal scales by which we see the aggregation of fish over a region, or even the length in time of a foraging trip of a top predator, then we fall more or less in the same scale of weeks, of days to weeks. So due to their strong energy and due to this temporal resonance with biological processes, the fine scales have a very strong structuring effect on the ocean biogeochemistry, on ecological fields. Here we see, for instance, that how easily physical features can be detected by looking at a biological field like chlorophyll concentration. And then due to their frontogenetic capacity, they are a regime for vertical and earthy exchange. So let's dive a bit more into the physics of the fine scale. We saw donc kind of an artistic view with the previous image. Here we see more in terms of physical processes with the fine scales that it's a term that now is used to put together the mesoscale and the submesoscale. They sit in between a regime where the rotation and the stratification is dominant, and a regime goes toward the three-dimensional. So that they are between the direct and the inverse energy cascade. So they play a key role in the flow of energy in the ocean, and they have this ephemeral nature that has been captured only in the recent decades. In fact, I will show here some extracts from relatively old papers. Let's say it was the blind period of the fine scales in which satellite images that were detected by in situ experiment only. So this is a paper from the 70s that looked at the large scale circulation. Then the authors they discovered that when they made the measurements of the Gulf Stream, there were some intermittent strong behaviors in terms of kinetic energy. Then the size of the features that were detected were of the order of a few hundreds of kilometers. Now we know that probably were rings, and that there are even smaller features that are there. And then we see that for these scales, the order of time for the lifetime is the order of a few months. And it was evocated the fact that this feature, this transient behavior, can play an important role in the general circulation. Another paper still in 70s identified this eddy energy that was even stronger than the background, the large scale circulation. And then at the end of the 70s, beginning of the 80s, the first remote sensing detection of ocean feature. Here we see a thermal image that started to draw this liquid landscape that we know now much better with the all satellite missions that have been launched together. So this was the case of one of the first remote sensed sea surface temperature. And then a few years later, there is of Nimbus 7 coastal zone color scanner that showed how the defined scales play indeed a key role on biology, in the sense that it emerged how the presence of filaments could structure also the phytoplanktonic distribution with potential impact over the trophic web. And so an interest about the managing of fisheries, over which we will come back in a few minutes. So more and more satellites have been added to our capacity of observing the defined scales, and with the computation capacity that went higher in the more recent years. So I would say that in terms of the challenges for defining scales, that can be probably two. Try to move away this bar. There are probably two classes of problems that can be addressed. One is in terms of physics, about energy cascade, heat material transport, fluxes, covalent exchange that I want to discuss much today. Here I just show an example about the latest generation of Earth system models that are used for the IPCC reports, which with the red box, their horizontal resolution in terms of degrees. So we see that most of them have a resolution of 1 degree. Some can go a bit smaller, but for sure they have not the possibility of resolving the defined scales, so that fine scale dynamics must be parameterized so the errors of the models in terms of energy flow can be constrained. So this is one of the key issues related to define scale. The second one that I will address more today is about biogeochemistry and marine ecology. If we look at what we know about the defined scales, there are some recent reviews in recent years. In this one, there's been a kind of a classification of our knowledge on defined scales in 3 categories. with the fact that the fine scales can move around, redistribute water patches, and this is what is called here passive processes. Then, because of their frontogenesis capacity, and in turn the possibility of inducing vertical motion, they can modulate nutrient upwelling and downwelling of organic matters that we know in most of their mechanisms, especially thanks to models. And then the last part in which there has been now a growing number of observations about the association of distribution of high trophic levels to define scales based on mechanisms that are largely unknown. So let me show you some examples of the fine scale dynamics in this regard. And before going to that, let's remember that we are living in a very special period in terms of open ocean ecology, because there have been some ambitious objectives that have been set at the political level, in particular an action for covering the surface of the ocean of marine protected areas, so that the 30% of its surface will be set for 2030. Considering that we are at about, depending on how marine protected areas are computed in terms of their protection level, from 3 to 7%, this means that a huge part of the ocean will have to be chosen for protection. And for doing this, we need to better understand its underlying mechanisms, especially at the fine scales, which are important for ecology, but are also the scales that are accidentally important for managing, for creating a managing plan inside a marine protected area. So I mentioned here in particular, you probably heard about the Biodiversity Beyond National Jurisdiction Treaty that has been approved in March this year, and that is a legally binding agreement for protecting the high seas, so outside the economic exclusive zone, that will open the way to the creation and the managing of open ocean marine protected areas. Okay, so this was to motivate you to listen to this talk, even if there is no much air in it, but there are some issues that are still of actuality. And now I speak a bit about the Lagrangian approach. So the Lagrangian approach tries to capture this part of the fine scale. And here we see an example, a conceptual idea of why sub-processes needs a Lagrangian view. Supposed to be along a current or a jet. Imagine that there are some nutrients that are upwelled in some region. Then the water is enriched as it passes over the seamount, and the processes for the trophic web start to be activated. So we start to have some phytoplankton to the source, and then after we have some aggregation of zooplankton. Now, since sometimes is needed from a phytoplankton to zooplankton compartment in terms of flow of biomass, this lag in time due to the current is transformed in a distance in space, so that it is not surprising that many predators actually forage in the open ocean far from nutrient sources. This is just because they don't eat nitrate and phosphate, but they eat a byproduct of that after a period for biomass to flow inside the web. Okay, so this is a conceptual view. This is an example of a work that we did some years ago, in which in the Southern Ocean we considered the possible effect of seamounts in a Lagrangian view. Considering, you see that this is latitude and longitude, so this plume of chlorophyll, it's actually quite extended in space. Then you see that in the top panel, potential sources of nutrient are marked in red as sea mounts. And then a Lagrangian model is used trajectories and looking at the far field effect of the nutrient sources downstream in respect to the Antarctic circumpolar current. So you see here that there's a strong relation of the model, the pattern that is reconstructed by the model, and the chlorophyll plume that extends downstream eastward from the seamounts. What is interesting is that this is also a known region of for foraging for several species of elephant seals, fur seals, and penguins, which are heading toward the region marked in red. And then you see the far field effect, the Lagrangian effect that I was showing before, in the sense that the region that is targeted by these top predators is not close to the seamounts, but is downstream to them in the zone where the Lagrangian model identifies some old water, where I forgot to say, the color that is in the top panel correspond actually to the time in day that the water parcel from when the water parcel has left the nutrient source. So for instance, here we are in a kind of dark gray patch. It means that a patch here has left a region close to a seamount 60 days before, which is a reasonable time for enriching this parcel with higher trophic levels. Now, if we zoom in the Southern Ocean and we look at fine scale features and now the behavior of a single animal, in this case an elephant seal, it is interesting to look at the relation between fronts foraging activity. So in gray here, you see the Lyapunov exponent, which is derived from altimetry, and is a measure of frontogenesis. So white lines are lines where steering create contrast by making a confluence of water masses that are coming far away. Then in violet, you see the trajectory of the elephant seals and of an elephant seal, and the color is darker depending on the number of prey items that the elephant seal encounter. Then you see that there are not so many. Then they suddenly go up when the elephant seal is at the frontier of a mesoscale eddy. Then you see that the value goes down at the moment in which the elephant seal leave the area. So you see here a clear association of a frontal feature with the foraging behavior of an animal. Interestingly, in this case, we found that the animal actually does not swim around the eddy, but is actually advected, entrained by the current around the eddy. In the paper, we speak about the planktonic behavior of the elephant seal, because basically the elephant seal is just going down inside the same water masses and is moving together with the water mass around the eddy. And then you see that we could do this analysis as usual. It's the best case that is shown because the eddy here is quite large. It's about 150 kilometers, and it's much more good canvas for finer scale features. This is something we'll come back with SWOT. Other examples, there are not only animals in environment, but people trying to fish. And to finish this part, I will show very quickly another example of the extension of a chlorophyll in this. This is downstream of Kerguelen Island in the Southern Ocean, which has been reconstructed with the Lagrangian method that put in evidence the recirculation feature close to the island. So an aging water mass that can be profitable for foraging. We cross that with the areas that are targeted by several species of top predator, and then you see that there are several species that are going in the same place, and then we had the satisfaction of participating to the working group for the creation of these marine protected areas, and this part was put in no-take. Then obviously it's also a zone where there is no commercial fishing. So politicians are very happy of finding an interesting ecological feature that has no fishing interest. But if we forget about this part, we are anyway very happy to see it there in the managing plan. I think I can switch to SWOT. In order to understand what SWOT can bring to the field of a fine scale, it's important to remind quickly how altimetry works today. So traditional altimetry has another instrument pointing toward the center of the Earth, and the distance between the satellite and the target, the surface of the water, is obtained by estimating the round trip of a radar signal. So we have a constellation of altimetric satellites that is a complex grid. And then when you use altimetry, you're not using this product, but you use a smooth map usually that comes from an interpolation. Interpolation means that there is a loss in resolution. So something like from this net of track this to convex diagnostics, for instance, Lyapunov exponents. Okay, could you hear me? There's been a loss. Hello? Can you hear me?
Yes, we can
Yes.
hear you. It's a bit
Okay.
broken up, but it's okay. Go on.
Okay. So these are the advantage and the locks of conventional altimetry. The advantage of being unpaired by clouds, and the fact that features up to about 100 kilometers can be detected. So limitations are for scales before, for coastal areas, and obviously that is by the geostrophic approximation. So is this enough or not? If you take a model that has other solution, in the panel down here, we made a reconstruction with the same algorithm that used for altimetry. So with a virtual constellation of satellites, we computed the Lyapunov exponents, and in the top, with the full resolution of the model. So you see that there are many, many features that are missing. The shape is different. Close to the coast, the situation is completely different from the one that current altimetries provide new us with reality. And so this was one of the reasons for SWOT that has another instrument in the middle, but also a wide SWOT instrument on the two sides that allow to take for the first time two-dimensional images of the ocean surface. So if we compare conventional altimetry with SWOT, then we have a gain of about 1 order of magnitude. If we were limited to 70, 100 kilometers, now we are limited more in the scale of 1 to 10 kilometers.
I think
We can access coastal area.
your slides are not changing.
Sorry?
We've
Oh,
been
no?
seeing the same slide when I think you're talking past this slide. Something's wrong.
You might just try
Okay, so there should be
stopping
a slide
sharing
with
and resharing.
conventional.
It might have lost its connection.
Oh, your screen? Yeah,
We're on a slide that shows 3 panels, a long track
your
processing,
screen
2D
sharing
mapping.
is paused. It says,
Yeah.
okay, I reshare the thing. Sorry about that.
No worries.
So here you should see conventional altimetry on
Yes,
the
perfect.
top and the SWOT on the bottom right. So these are the differences and in between the two different types of technology. To resume, SWOT has been launched. Just a second that I move away this.
Would you like to switch your video off? Because I think we have a bandwidth issue. Because we can see your lips move, but you can't hear anything. Okay, try again. We can't hear you.
Can you hear me?
Yes, you
Yes.
can now. Thank you.
Okay. Or maybe I don't know. Anyway, so this is the situation now in which the coverage of SWOT is global. There has been a first part in which SWOT was in a closed orbit with a repetition of 1 day, in which we have organized this beautiful experiment that has involved about 30 different groups for the ocean. And I conclude very quickly with an example from the BioSWOT MED. So we've been lucky because just before the cruise, we had a series of very nice Sentinel-3 images. So we targeted these features that you see about 100 kilometers northeast of Minorca. We are in the Mediterranean Sea. Then you see how quickly this feature evolves. This is how conventional altimetry see this area. So there is possibly a front, but clearly not all the richness that we see in chlorophyll. And this is the image that we had in near real time from SWOT. So I show again the difference for people that are not used to SWOT, and then you see that the gain is unbelievable. It's really like comparing a high resolution model with a low resolution one. This allowed us to go inside this anticyclone that is typical for the Mediterranean Sea, but it's a type of feature that have not been studied before because it is invisible. These kind of features are simply invisible on natural altimetry. This is the strategy for our campaign, in which we had a lot of underway instruments and biology, from cytometry to a zoo glider up to genomics. We had a sea star that flew with us, looking for the ageostrophic components. A lot of drifters that have been released to characterize the fine scale circulation. And just to finish, small example of NRC interaction in the sense of internal waves that have been excited inside our small feature. So this is not completely obvious because the eddy was much smaller than some other eddies that exist also in the Mediterranean Sea, like for instance, Algerian meanders of the Algerian current. And a big storm could also, we could imagine that could destroy an eddy. But in this case, we saw that inertial waves were trapped and generated internal waves, and mixing was then created when the waves decayed. So we are now looking at nutrient fluxes and the response, for instance, in terms of metatranscriptomics to the fast response of the phytoplanktonic community. So conclusion. So I started with the fine scales in terms of current water parcels, a bit like land parcels in a terrestrial landscape. The specificity that they have a short lifetime that is in resonance with biological processes, and that that create strong contrast due to their strong kinetic energy. Part of the variability of the fine scale, and especially their, let's say, memory effect during their travel with the current, can be captured by Lagrangian analysis that estimate transport and steering. So I show some applications of Lagrangian methods, including the identification of nutrient pathways, the understanding of behavior of top predators, some application with conservation, and the possibility of detecting this feature in near real time, as we did during the SWOT campaigns. So conventional altimetry has been a great instrument and a very powerful tool. It had some limitation, especially in terms of resolution, and the fact that it had some biases close to the coast. Now SWOT has broken this barrier. We have maps that are 10 times better resolved with some data that are not hindered by the cost. So there are still some frontiers. Well, we have plenty of possibility of exploiting SWOT data as we started to do. But if we think to future satellite missions, that anyway it takes many years before they are launched, then things that are still open are the non-geostrophic currents and possibly the fact of co-localizing in space fine scale currents together with some other properties. And I finished. Thank you very much. So I've been a bit long. Well, we started 5 minutes later. So
Thank you, Francesco.
Thank you.
Thank you, Francesco. That was very interesting. And apologies for not being here to welcome you earlier, but we're just a week before the Sister UCM, so we were running a bit late. Apologies. Yes. Anyway, that was very interesting. Thank you for this presentation. So, do we have any questions for Francesco? I will start by asking about the fine scale. You kind of define now fine scales are being mesoscale and submesoscale. I mean, typically they've always been discussed kind of separately. So, you know, I'm making the case for, you know, high resolution 1 kilometer 1 day kind of observations. Is there still an argument to separate this sort of scales in this way, as you know, short small scale features very quickly and large scale features very more slowly? Or
I think that is very practical to think of them as a separate, because that helps us to write equations, to address processes. But in the ocean, we see that they are not really separated. When we look at an image of surface chlorophyll concentration, for instance, is very far from being a picture of round spot in some regions and only filaments in the other. And it's not a matter of zooming in on or out. We see that there is a smooth transition in between regions that are more recirculating and regions that are more filamenting. So to me, if we have to think in terms of coupled processes of tracers, it's not really acceptable to cut in between mesoscale and submesoscale processes, which is a good argument for you, because it advocates that sea surface height is not enough to characterize the coupling between physics and biology, because of the non-geostrophic component, which becomes dominant as we go.
Which I hope you will be able to show to strengthen our case for our mission. But there's already a lot of evidence indeed for those ageostrophic motions being very important. Sarah, you have your hand up.
Yeah, this was really interesting. And I guess I was thinking from the air-sea flux standpoint, because I think there is a lot of overlap, but everything in the upper ocean depends on air-sea fluxes. But I was wondering what happens, I guess, what the role of, you have the potential to address the biological pump in looking at biological productivity. And I wondered how much you thought about that role of really trying to assess CO2 uptake, and how much that might be modulated by the presence of or absence of eddies and frontal features.
So I can answer in terms of SWOT, in the sense that we thought what SWOT can add in that regard. And I think that one of the key questions is that when you have in situ data, you want to colocalize as best as possible with frontal features or with physical features, and with ultimately even for large eddies, the structure is smoothed and is much rounder than in reality, because anyway the maps come from interpolation. With SWOT, the fact that the feature is observed with the two-dimensional SWOT allows you to colocalize very precisely and in situ estimation of carbon fluxes, nutrient export of biomass, very precisely with the part of the physical feature that the measurement is in. Then the other advantage is that with SWOT we should be able to reevaluate at the global scale the intensity of vertical velocities that still remain one of the graal of oceanography. Eric knows about it since some years. And the fact of doing targeted campaigns, like for instance SMOD, I think is one of the best examples about the link between sea surface, high resolution sea surface height and vertical velocities. At some point could be able to provide a method to map vertical velocities from space.
Okay, thank you.
Thanks.
Any more questions? So I was very interested that, you know, SWOT went into this one-day repeat for 90 days or so. Do you have any feel of how you're going to use this quite unique kind of sampling to understand some of the processes that you're looking at? Is there, you know, what's the value of that one-day repeat for what you're doing?
Well, there was a trade-off with the SWOT mission in terms of coverage and temporal resolution. So of course it is very tempting to have a mission that is high resolution and there's a global coverage. If you propose a mission that can see only a few lines scattered over the globe, I think that's not very attractive. But the fast sampling phase has the possibility of looking the fast temporal variability of the sea surface height and serve as to calibrate and validate the products that are made by the dynamical interpolation that still is necessary to put together all of the global SWOT of SWOT. And then, so there's this was the first motivation for the fast sampling phase in terms of CalVal. And then the second one is the one that has been exploited by the many experiments, is that the SWOT are about the sides of a typical region that is explored with a campaign, because they mix about 120 kilometers. So it's a reasonable size for making campaigns. And in that case, having on a region around the ship one image per day or even two in a crossover, it's a great advantage to look at the temporal, well, the life cycle of the features that are explored in terms of balanced motion, and also in terms of internal waves that are strongly aliased during this global phase.
Okay, thank you. I see Eric has his hand up. Please, Eric.
Yeah, hi. That was a fun talk. One of the things that one would like to do with SWOT is be able to do these Lagrangian coherent structures sort of calculations on a global scale and on a much finer scale.
Yes.
But one of the one of the issues there is that the repeat time is really long. It seems longer than the time scale of the small scale Lagrangian features. So have you thought about how SWOT analyses can overcome that issue?
Yeah, well, we are also trying to do the same. So I didn't show some images, and we did the calculation over for the fast sampling phase. So we embedded the SWOT over the standard UX map, so that when the trajectories goes out from the SWOT, they still have something to be propagated. And you see something very similar to what we expect from models, in the sense that you find that in terms of Yapon exponents, they are about twice stronger than the calculation from UX. And then you see a tangle of very thin features. So now level 3 products for the fast sampling phase are a bit noisy for being ideal for this calculation, but the new version should come very quickly. And for the global scale, that's very tempting as well. I think that the interpolation that has been used to create one-day global maps of SWOT is quite reasonable, in the sense that it's not just an interpolation, but what they do, it's a kind of a dynamical assimilation of the passage of SWOT, so that at the end you really see that the small features that are seen over a SWOT can propagate over the maps. The difference in respect to the standard UX algorithms is really impressive for the propagation. In one case, you see small eddies that propagate. In the other case, you see small eddies that are disappearing and reappearing. So I think that the calculation at the global scale should not be very problematic, especially if it is validated by the calculation for the fast sampling phase. But I'm revealing you our work.
We'll find out. We'll find out how well this works. That's cool.
We're all in it together, Francesco. Don't worry.
Yeah.
Rick, you have a question.
Yeah, hi, thanks. Just a quick question, Francesco, about a model of biological processes, a basic model, or really the simplest possible model, because that's where we don't, I think one of your slides showed that we don't have much knowledge of how biology reacts, all trophic levels react to these features that we can see, these physical features. So I'm wondering if it would be correct to say that the simplest model of biology would be the one where one wants to eat and not be eaten.
I think it's very one of the interests of having a 10 times higher resolution is that we can test some simple ideas with real data. Before we can see something, for instance, in biologging, in terms of for testing some hypotheses on the behavior of some species. But there is a lot of noise in the Mediterranean Sea, for instance. So there is hardly anything that can be done because most of the eddies are invisible to altimetry. So your idea is probably very tempting, and is something that could be tested with much better with SWOT data than what was possible before with altimetry, and it may work for in some cases. Maybe it will work. I doubt that it will understand everything for all species in the whole ocean, but could be one of the explanation for some
Great,
conditions.
thank you.
Thank you very much. I see no other hands up. This is your chance. We're coming to the end of the hour, so unless there's anything else. Mohammed has a question. Go on.
Okay, thank you. I don't have a question actually, but I have a comment.
Okay.
So thank you for that. It was an excellent presentation. Actually, it's out of my the biology thing is out of my expertise, but I found it useful to mention that during the BioSWOT campaign, I managed to acquire a very good data set of radar subconstellation mission RCM C-band SAR with one image every day. So at a resolution of 30 meters. So it seems to me that some features that we could extract from these images would be useful to them to what it's not open publicly for the data, but I would be interested. It would be a good idea, Francesco, to exchange some emails probably off the line to see how this data could be, useful, especially for the field observation that you conducted. Thanks.
Yeah, with pleasure.
Yes, just
Yes.
to clarify, Mohammed is on the MAG for C-Star, and we have
Yeah,
also
I understand, yeah.
collaborated for the Oscar flights, which you have mentioned happened during the BioSWOT campaign. So yeah, there's a lot more data there and stuff to look at. There's also some amazing information in the SWOT sigma zero images that we get and how those compare to what we see in other SAR images. So there's a lot of work there to be done. So I know that this webinar series is very strict on finishing exactly on time. So we don't have time for any more questions. I will just say one word, which is next week we have no seminar, no webinar, because it's the user consultation meeting for Earth Explorer 11. So we decided not to have a webinar. It's literally a week from now, so it's all getting a little bit scary. I believe that ESA is providing access through their ESA web TV or something. If you go on the UCM webpage, it explains it. And I think they're still working on a way to make sure people who are viewing that can actually ask questions. But there is still a possibility that people who are viewing may not be able to ask questions. But we keep our fingers crossed that they do something sensible.
Yeah,
so I hope to see. Yeah,
keep
sorry.
us posted, Christine, as you find out what's available.
Yes. Well, they don't tell me, I have to look at the website like everyone else. They don't tell me anything. So I hope to see, many of you kind of online and make your voices heard to make sure that, we have evidence that the community is very much behind C-Star. And yeah. And we have come to the end. Sarah, do you want to say the final words since you're...
No, I'm going to stop recording. Thank you very much.
Okay.
Thank you so much for the
much.
invitation.